Today we'll discuss antimicrobial drugs. So Alexander Fleming in 1928 discovered penicillin, and he was actually studying Staph aureus, and he noticed that there's some contamination in the plate with mold, and the area around the mold here, which is clear, showed that something was being produced. So that started a whole lot of research into different types of antibiotics. So we have two main classes, guys, and those are bacteriostatic, and static means to inhibit, so that those types of drugs prevent bacteria from growing, so they inhibit them. Cidal means to kill, so bactericidal will actually kill the bacteria. So there's 5 different places that can be targeted. Cell wall, for instance, plasma membrane, nucleic acids, protein synthesis, and folic acid synthesis in the cell. So most bacteria have the cell walls that consist of peptidoglycan. So there are some drugs, like penicillin for instance, that will block peptidoglycan production, and they do that by interfering with what's called transpeptidation. And what that does, guys, is it links the layers of peptidoglycan together. So if you stop that from happening, then the cell wall is going to fall apart. So for your exam, please know these 5 classes of antibiotics and a little bit about each one. So in the cell wall inhibitor category, we have penicillin, and that inhibits cell wall by inhibiting the enzyme that forms peptide bridges, and that enzyme is called transpeptidase. Isoniazid will inhibit mycolic acid. Now, mycolic acid is found in tuberculosis, so we give that in combination with other antibiotics to treat tuberculosis. There are 3 protein synthesis inhibitors I'd like you to know. First one, streptomycin. It will bind to the 30S ribosome that causes misreading of messenger RNA. The next one, tetracycline, binds the 30S ribosome, and that will inhibit transfer RNA from binding to the A site of the ribosome. And the last one I'd like you to know is erythromycin. That binds the 50S ribosome, which will inhibit translocation of ribosomes. Now we have one particular class of inhibitors of essential metabolites that we call sulfonamides. So trimethoprim and sulfanilamide, those will inhibit folic acid synthesis. As far as inhibitors of nucleic acid synthesis, the 2 I would like you to know are ciprofloxacin, that inhibits the enzyme DNA gyrase, and rifamycin, that will inhibit RNA polymerase. For cell membrane damage, polymyxin. That will interact with membrane phospholipids, which distorts cell surface, and it will cause leakage of protein and nitrogenous bases from the microbe. Now, we have what are known as beta-lactam antimicrobials. Again, penicillin is an example of that. And basically, it binds to transpeptidase enzymes. When it comes to antiviral drugs, please know that they are classified into 5 main groups, depending on their activity. So some will block attachment of the virus. Some will stop it from penetrating. And again, the ones that infect us will penetrate by endocytosis. Uncoating, so once the virus is brought in, if you can't get that capsid removed, the virus will pass through the cell and not harm it. The 4th category is viral replication and assembly. So if the virus can't replicate itself, if it can't put itself together, it's not going to be able to harm us. And same thing, viral release. If that gets inhibited, eventually the virus is not going to be able to go on and infect other cells. As far as fungal infections go, please know the three main classes: azoles, alliamines, and polyenes. So these are all effective then in treating fungal infections. Now, one of the things that we have to look at is how susceptible microbes are to the drugs that we actually treat them for. So, for instance, over 2 million people will suffer from some kind of antimicrobial resistant infection, and 23,000 of these patients die due to the infection. So we have to take a look to see how susceptible microbes are to the antimicrobials that we are treating them with. So there's two tests that are done, Kirby-Bauer and the E-test, and the one I'd like to talk a little bit about is the Kirby-Bauer test. That's one that we always do in the micro lab course at SPC. So for the Kirby-Bauer disc diffusion, we use a specific type of agar called Mueller-Hinton agar. It's got a pH between 7.2, 7.4, and it's poured 4 millimeters thick. And that's to keep the diffusion of the antibiotics from spreading too far. So if you pour plates too thin, what happens is those zones get very big and run into each other. So the zone of inhibition then is the clear area around the disc where the bacteria are prevented from growing. So here's an example. The disc is the white little dot in the middle. The clear area is the zone of inhibition. So what you do then is measure this and compare to a standard reference chart to determine, is the bacteria sensitive or resistant to the antibiotic? So again, all antibiotics have different properties. So their zones of inhibition, as far as how effective they are, is going to be different. So again, if the bacteria is susceptible, the zone of inhibition falls in the susceptible range on that chart. If the bacteria is resistant, the zone of inhibition falls in the resistant range. And if it falls in the intermediate, that's a borderline response. So one thing I'd like you to keep in mind with intermediate responses, it's possible that the bacteria is becoming resistant to that antibiotic. And you would only want to use an intermediate if it's the last resort that you have. So you could adjust the dosage maybe, or the length of time. But it's always the last result if there's no other antibiotic that has any effect at all. Now, resistance occurs when the microbe is not affected by the drug. And resistant microbes are quite often called superbugs. So there are two types of antimicrobial resistance, intrinsic resistance and acquired resistance. For intrinsic resistance, please know it's a natural resistance to antimicrobial drugs. So that makes certain pathogens harder to eliminate because this is a natural thing that they're actually producing. Acquired resistance means that they get the resistance from some type of genetic mutation or by acquiring some type of resistance genes. So we have to be aware that in the environment and even within your body, if there's a way that the bacteria can uptake a gene, for instance, one that's in a plasmid, for instance, it will become resistant then to the antimicrobial drug. So please know that there are three main ways microbes evade antimicrobial drugs. One, they alter the drug's target. Secondly, they actually inactivate the drug. And lastly, they reduce the drug concentrations inside the cell by blocking the drug entry, or in some cases, they actually pump the drug right out of the cell. Now again, there's a lot of behaviors that can affect resistance. For instance, we know that livestock, for instance, are treated with antibiotics, so the microbes that they may have will become resistant. Also, manure could be contaminated with resistant bacteria. People fly around the world, but not all countries regulate antibiotic use. Not everybody takes their medication the way that they're supposed to. So if you don't take the whole prescription, guys, it's quite possible you're going to be starting to breed resistant types of bacteria. Also, if you are a healthcare worker, you're exposed to more resistant types of microbes than most normal people would be. So many patients, again, don't take all their doses. Or sometimes they save them and then they take old ones that are not as effective anymore. Also, what happens by doing that is you are making, believe it or not, the bacteria resistant to that antibiotic. Again, antibiotics are fed to our livestock. So there's about 26 million pounds of antibiotics used in animal feed. So the FDA banned a lot of antimicrobials used in human medicine to animal feed for growth promotion purposes. So we have to be careful with that. But again, there's a lot of what's called cross-contamination that can still occur anyway. 30% of oral antibacterial prescriptions are unwarranted. So what that means is that the wrong drug is used. Maybe the patient was misdiagnosed. Maybe the patient had a cold and they said, hey, I need a prescription here. And again, keep in mind that in other countries, you don't even have to have a prescription to get an antibiotic. Please take a screenshot of this. Now, something I hope you'll all find interesting. Healthcare workers and staff pick up a lot of resistant strains on their hands, scrubs, and personal electronic devices. So again, good hand washing. Wearing disposable protective covering is a good thing. Don't have your cell phone out and you don't have to worry. So what happens then in these cases is that the person can transfer to themselves or others in their community, their families, or to patients. So please note the last point here, guys. One study found that antibiotic-resistant bacteria could be isolated in 32% of sample clinical personnel's clothing and or their handheld electronic devices, i.e. cell phone, iPad, etc. So we have a lot of different resistant microbes to watch. CDC's keeping an eye on that, amongst other things at the moment. So again, please follow the proper care and way of doing things at your institution, guys, washing your hands again, etc. Patients need to make sure they're following the dosages that they have and store your medications properly in a cool, dry environment. There's not a lot of new antimicrobials out there, guys, because there's not a lot of money to be made, but there's a lot of other types of things that are coming up the pipeline, hopefully, to take care of this problem of antibiotic resistance.